After studying this module, you shall be able to understand about AFLP, that is Amplified Fragment Length Polymorphism Technique, and its uses, the principle and the steps involved in this technique. A novel DNA fingerprinting technique called AFLP, that is Amplified Fragment Length Polymorphism, is based on the selective PCR amplification of restriction fragments from a total digest of genomic DNA. This technique involves 3 steps. Number 1, restriction of the DNA and ligation of oligonucleotide adapters. Number 2, selective amplification of sets of restriction fragments. And number 3, gel analysis of the amplified fragments. PCR amplification of restriction fragments is achieved by using the adapter and restriction site sequence as target sites for primer annealing. The selective amplification is achieved by the use of primers that extend into the restriction fragments, amplifying only those fragments in which the primer extensions match the nucleotides flanking the restriction sites. Using this method, sets of restriction fragments may be visualized by PCR without any prior knowledge of the nucleotide sequence. The method allows the specific co-amplification of high numbers of restriction fragments. The number of fragments that can be analyzed simultaneously, however, is dependent on the resolution of the detection system. Typically, 50 to 100 restriction fragments are amplified and detected on denaturing polyacrylamide gels. The AFLP technique provides a very powerful DNA fingerprinting technique for DNAs of any origin or complexity. It is a technique used to detect polymorphisms in DNA when no information about the genome is known. Following restriction enzyme digestion of DNA, a subset of DNA fragments is selected for PCR amplification and visualization. A unique fingerprint is generated for a particular genome. AFLP, which was first developed for plant studies, is now used for a wide variety of genetic analysis applications. It is one of the tools scientists use to distinguish organisms based on their genomes. With this technique, even small amounts of genomic DNA can be used to produce DNA fingerprints that are highly specific to particular species. In order to understand how AFLP PCR works, it is important to understand what we mean when we use the term DNA fingerprinting. In short, DNA fingerprinting involves the generation of a set of distinct DNA fragments from a single DNA sample. These fragments are then used as a source of genotypic information. A wide range of techniques can be used to generate DNA fingerprinting data. All DNA fingerprinting methods study patterns that are associated with genetic markers. However, individual techniques differ in terms of the number and the type of genetic markers examined. For example, some approaches allow the examination of a marker at a single locus called single locus markers, whereas others allow the simultaneous investigation of multiple loci called multi-locus markers. Some approaches focus on codominant markers which provide information about both alleles present at a given locus. In contrast, other techniques are concerned with dominant markers which only report the presence or absence of a given allele and cannot provide information about whether an individual is homozygous for that allele. The oldest method used in DNA fingerprinting studies is restriction fragment length polymorphism, or the Rflp analysis. Rflp analysis has been widely used to identify genes linked to several Mendelian, that is, single gene diseases such as Huntington's disease. This approach detects differences in DNA fragment lengths due to the presence or absence of a restriction enzyme site or due to an insertion or deletion that occurs between two restriction enzyme sites. As opposed to Rflp analysis, the second DNA fingerprinting technique focuses on microsatellite regions of the genome that contain simple sequence repeats, that is SSRs, which are short stretches of 2 to 6 nucleotides that are repeated multiple times. Most often, SSRs consist of 2 or 3 nucleotides, for example, CA, CA, CA repeated many times. The number of SSRs within a given microsatellite region of the genome often varies among individuals. Because of these differences, we can use PCR-based approaches with primers that flank the microsatellite region to determine the length of the SSR. The size of the resulting PCR products is determined using high-resolution gel electrophoresis. This approach requires knowledge of the genome sequence to design the primers, but it can be used to detect multiple alleles for a given microsatellite region. Yet another approach to DNA fingerprinting, called the random amplified polymorphic DNA or the RAPD analysis, uses PCR primer sets designed to randomly amplify DNA fragments scattered throughout the genome. RAPD is a multi-locus approach that uses low stringency PCR conditions to permit promiscuous amplification of genomic regions. It can be carried out in the absence of genome sequence data, but the low stringency conditions sometimes provide challenges when trying to reproduce or interpret results. Scientists are now increasingly turning to Aflp PCR in their DNA fingerprinting efforts. Aflp PCR uses many of the same steps as Rflp, SSR and RAPD, including restriction enzyme digestion of genomic DNA, PCR and electrophoretic separation of DNA fragments. However, this method includes additional steps that permit high resolution interrogation of the entire genome, and it yields highly specific reproducible genotypic data. Although AFLP can only be used to study dominant genetic markers, it does not require any previous knowledge of the genome sequence. Principle: the AFLP technique is based on the amplification of subsets of genomic restriction fragments using PCR. DNA is cut with restriction enzymes, and double-stranded or DS adapters are ligated to the ends of the DNA fragments to generate template DNA for amplification. The sequence of the adapters found and the adjacent restriction location work as primer binding sites for amplification of the restriction fragments. The nucleotides are carefully put at the 3' ends of the PCR primers, which as a result can only synthesize primer DNA from a subset of the restriction sites. Only those restriction fragments in which the nucleotides adjoining the restriction site match the selective nucleotides will be amplified. The restriction fragments for amplification are generated by two restriction enzymes, an uncommon cutter and a common cutter. The AFLP procedure results in predominant amplification of those restriction fragments which have an uncommon cutter sequence on one end and a common cutter sequence on the other end. The basis for using two restriction enzymes is the following. Number 1, the common cutter will produce small DNA fragments which will amplify well and are in the appropriate size range for the purpose of separation on gels. Number 2, the quantity of the fragments to be amplified is reduced by the use of the infrequent cutter, since only some cutter fragments are amplified. This limits the quantity of selective nucleotides needed for selective amplification. Number 3, the application of two restriction enzyme markers makes it probable to label one strand of the DS-PCR products, which prevents the occurrence of doublets on the gels because of unequal movement of the two strands of the amplified fragments. Number 4, using two unlike restriction enzymes gives the highest flexibility in tuning the number of fragments to be amplified. Number 5, large number of different fingerprints can be generated by the various combinations of low number of primers. Methodology: the technique of AFLP was first described by researcher Peter Voss and his colleagues in 1995, Voss et al. 1995. This technique involves 5 major steps. Step 1, preparing the AFLP template. Step 2, ligation reaction with restriction fragments and adapters. Step 3, selective PCR amplification. Step 4, electrophoretic separation of amplified DNA fragments. And step 5, analysis. Taking these steps in detail, step 1, preparing the AFLP template. AFLP analysis starts with a sample of genomic DNA being isolated and purified from an individual. After the genomic DNA is obtained, it is digested with a pair of restriction enzymes, often MSC1 and EcoR1. MSC1 recognizes 5' TTAA 3' and cleaves after the first 5' T, whereas EcoR1 recognizes 5' GAA TTC 3' and cleaves after the 5' G. The genomic DNA is incubated with these enzymes for a period long enough to permit complete digestion of the DNA into restriction fragments. MSC1 and EcoR1 generate DNA fragments with 5' overhangs, that is 5' TA 3' and 5' AATT 3' respectively, that are distinct from each other and are non-complementary. Step 2. Ligation reaction with restriction fragments and adapters. In order to make use of the fragments of genomic DNA generated by MSC1 and EcoR1 digestion, researchers needed a method that would allow them to generate a unique type of DNA fingerprint. For this, they developed an MSC1 adapter and an EcoR1 adapter, each of which is a short double-stranded DNA molecule with a 5' overhang that is complementary to the 5' overhang generated by MSC1, that is 5' TA3' and EcoR1, that is 5' AATT3' respectively. Rather than containing a 3' A after the 5' TA3' overhang, the MSC1 adapter contains a 3' C which destroys the MSC1 recognition site once ligated. Similarly, rather than containing a 3' C after the 5' AATT3' overhang, the EcoR1 adapter contains a 3' G which destroys the EcoR1 recognition site once ligated. The MSC1 and EcoR1 adapters each contain their own specific DNA sequence, typically between 19 and 22 base pairs in length, that is located upstream of their respective 5' overhangs. The aforementioned ligation reaction involves incubation of the MSC1 and EcoR1 digested genomic DNA together with the two adapters and with DNA ligase, which is an enzyme that covalently links the adapters with their respective complementary 5' ends. MSC1 and EcoR1 restriction enzymes can also be added to this ligation reaction, as they will prevent relegation between EcoR1 and MSCI genomic fragments and will not recognize the ligated adapter genomic DNA fragments. Step 3. Selective PCR amplification. After ligating the genomic DNA fragments with their corresponding adapters, researchers are left with a series of DNA fragments that consist of the specific MSC1 adapter associated sequence, the destroyed MSC1 site, a genomic DNA fragment, the destroyed EcoR1 site, and the specific EcoR1 adapter associated sequence. Therefore, each unique genomic DNA fragment is between the MSC1 adapter and the EcoR1 adapter and their associated unique DNA sequences. Although the MSC1 and EcoR1 adapter ligated ends of each DNA fragment will be identical, the sequences of the genomic DNA fragments, which begin just after the original MSC1 and EcoR1 sites, will differ. If the PCR reaction are carried out using primers corresponding to the MSC1 and EcoR1 adapter sequences, every single genomic DNA fragment is amplified and they would therefore be faced with an indecipherable set of DNA fragments which are obtained. Thus, in order to selectively amplify a smaller number of genomic DNA fragments, researchers use primer sets that are complementary to the MSC1 or EcoR1 adapter sequences starting at their 5' ends, but that add up to 3 unique nucleotides following the end of the original MSC1 or EcoR1 recognition site. The addition of only a small number of base pairs called selective nucleotides at the end of each primer permits the amplification of a subset of genomic DNA fragments. The more complex the genome being investigated, the more selective nucleotides researchers add to the primers. Some simpler genomes might not require any selective nucleotides at all. As the number of nucleotides added to the 3' end of each primer is increased, the selectivity of the primer also increases. After all, more genomic DNA fragments will be amplified by a primer that contains a single unique base at its 3' end compared to a primer that contains 3 unique bases at its 3' end. The primer design does not require any previous knowledge of the genome under study. Rather, researchers simply set up PCR reactions using a variety of primer sets, each of which consists of one MSC1 associated primer and one EcoR1 associated primer. Aflp PCR reactions are carried out under stringent conditions, permitting only the selective amplification of those genomic DNA fragments that are perfectly complementary to the 3' ends of the primer sequences. The stringent PCR conditions employed in Aflp PCR reactions lead to highly reproducible results that are readily comparable among different samples. Step 4. Electrophoretic separation of amplified DNA fragments. The PCR reactions proceeded at the third stage of the Aflp PCR procedure will at the same time amplify a very wide range of DNA fragment sizes, which is based on the length of original MS1 EcoR1 genomic DNA fragment ligated to the MSC1 and EcoR1 adapters. In order to determine the Aflp PCR derived DNA fingerprint, researchers must be able to identify and characterize the set of DNA fragments produced in a given PCR reaction. Thus, in most cases, one of the two PCR primers is radioactively or fluorescently labeled, typically the EcoR1 primer, which leads to the production of a labeled PCR product that can be easily detected. Researchers also use high resolution electrophoresis to separate the DNA fragments based on their size and overall negative charge. Large fragments will move more slowly and small fragments will move more rapidly when subjected to an electrical field. Step 5: Analysis. Following Aflp PCR, researchers must analyze the resulting DNA banding patterns to determine the DNA fingerprint associated with a given sample. Depending on the complexity of the banding patterns, researchers can manually determine band sizes or they can turn to automated approaches. Because Aflp PCR can only detect dominant genetic markers, it cannot report whether an individual is homozygous for a given marker. However, this technique does not permit the simultaneous amplification of multiple genomic DNA fragments, that is, often between 50 and 100 fragments within a single PCR reaction with high specificity and reproducibility in the absence of any genomic sequence data. The Aflp method is purely based on the selective PCR amplification of restriction fragments. This technique consists of 3 steps. Number 1, restriction of the DNA and ligation of oligonucleotide adapters. Number 2, selective amplification of groups of restriction fragments. And number 3, gel analysis of the amplified fragments. In this, the cellular DNA is digested by restriction enzymes followed by ligation of half site specific restriction adapters at restriction fragment site. With the help of 2 PCR primers with the respective adapter and restriction site specific sequences, the amplification of the selected fragment site is carried out. Then these fragments are separated by applying electrophoresis technique followed by visualization of the band pattern. PCR amplification of the restriction fragments is attained by the usage of the adapter and restriction site sequence as target position for primer annealing. The selective amplification is attained with the help of usage of the primers that spread out into the restriction fragments, amplifying specifically only those precise fragments in which the primer extensions match the nucleotides adjoining the restriction sites. The number of fragments that has to be analyzed simultaneously on the other hand, depends on the resolution of the detection system. Typically, 50 to 100 restriction fragments are amplified and identified on denaturing polyacrylamide gels. The AFLP system provides a unique and very influential DNA fingerprinting method for DNAs of any origin or complexity.